Thank you very much. My name is Rachel Melamed, and I'm actually at UMass Lowell. I don't know why I have this old affiliation up, but it's my fault. And I'm going to talk today about our method for discovering drugs' effects on phenotypes, so new drug indications, new uses of drugs, by linking drugs' effects on phenotypes that are known with drugs' effects on molecular signals. Across hundreds of common drugs and common phenotypes. And this work is recently published in Bioinformatics Advances, so you can check that out. I'll share this QR code a couple more times. So one of the big goals of our lab is to discover new effects of common drugs using data. So if we want to ask if a particular drug impacts a particular disease, there's a number of types of data that we can use. So one is human level data, such as epidemiological studies, where we can look at people who take a drug and ask, do they have a changed risk of developing a disease, and also use information of known indications of a drug. And that's the topic of a presentation our lab's doing tomorrow at Transmed at 3:20. And it's also a preprint that's available if you're interested in checking that out. But another type of data that we can use is molecular level and genetics level data. So that is, we have information on the genes and pathways altered by a drug and the genes that are involved in a disease. And we can use that to investigate if there's some connection between these two entities that could explain why a drug might impact a disease. And looking at the genetic similarity between diseases for drug repurposing is the topic that my student is presenting at Transmed, actually right after this. So you can also watch the recording. That's in room 519 as well. And my other student, Katie, is presenting a poster tomorrow in the poster session about using drug similarity for drug repurposing. But for this project, we wanted to see, can we make a model that integrates both the human level data, this epidemiological level data, and the genetic level data at once to discover both drug effects on disease, but also the biology of drug effects on disease. So we want to understand not just does it affect it, but why does it affect it? So here we propose this proposed model that we call Draphnet in the end. So we propose that drugs affect some sort of cellular processes, and those are downstream impact the genes that later result in a disease arising. So to make this a little bit more concrete, we have data that profiles for any particular drug the different physiological effects, the different cellular processes impacted by that drug. So some examples of this would be like, does this drug impact androgen agonist activity? Does it affect the MAP kinase pathway? That's data that already exists that we can profile a drug with. And similarly, for any particular phenotype, we can use GWAS, gene-based results, to say which disease genes, and I'll just call them disease genes, are involved in that particular phenotype. And so a third source of data, in addition to those other two data, is, like I said, drug indication. So does a drug actually impact a phenotype by treating that phenotype? That's called a drug indication. So if we have these three sources of data, we propose that by bridging the two molecular sources of data, we can predict drug indications. So we're trying to learn the connections between these cellular processes and the genes involved in disease. And that's in the form of a weighted network. So how can we learn this weighted bipartite network connecting cellular these genes? And so what we're doing is the simplest thing we could think of. Maybe I'm the only person at ISMB still doing linear models, but I'm doing it. So we're just learning a simple linear model. So we're taking ToxCast data, which, like I said, is profiling all of these diverse cellular endpoints, where we can represent a drug with a vector, just saying how much the drug impacts each of these 1,000 or so endpoints. And similarly, for a phenotype, we can use gene-based GWAS results to again formulate a vector saying, does the drug impact each one of these genes? So like maybe all of the common genes that are involved with any of these phenotypes. And then our model boils down to learning this W matrix that connects these two other vectors. And in theory, we can multiply these three entities together, dW times P. And if we learn the right W, we should be able to predict whether that drug impacts that phenotype, the probability of that. And so we just train the W to predict data on drug indications that's stored in CIDR. So we're training this across 70,000 pairs of drugs and phenotypes, where we have for each one of those pairs is just, does that drug treat that phenotype or no? Yes or no? So across all of those pairs of phenotypes, we have one matrix W that we're learning consistent across all of them. And that is what we call the drug phenotype network W. And so this boils down to a sparse regularized regression, like I said, a linear model. So that's potentially very simple, but does it work? That's really the question. Does this actually work? So we do a whole bunch of evaluations to say, does that work, by saying, how well does this model predict drug effects? So what do we mean by drug effects? So there's a number of ways to think about that. First is the drug effects on phenotypes. That means, are we able to predict drug indications? So again, we can just multiply these three things together, get our predicted probability for every drug of whether or not it treats that phenotype. And we can compare that to held out drugs and say, how well are we able to predict that? And there's a bunch more details in the paper, but we compare that to a bunch of different benchmarks. And we're able to show that actually we do have pretty good performance on that task. But that's just one task. And I would say that's like the less interesting task that we're able to do here. Because what I find really exciting is that we're actually able to predict drug disease gene effects. So what I mean is that we have this matrix D connecting each drug to its effects on endpoints. That's our data. And then we have this learned model W connecting each of these endpoints to disease genes. And then if we just multiply these two things together, we can get, does each drug affect each of these particular genes that is then involved in disease? So we get this matrix connecting each drug to the set of genes that are involved in the disease. And we call this the drug disease genome mapping, or the drug disease genome matrix. So the next question, of course, is, are we actually learning something reasonable? Because if so, I think that's kind of exciting. We could look up any drug we're interested in and say, which of the genes that are involved in disease does this drug actually affect? So that could be a great resource. So what we do is evaluate whether this is learning new biology by comparing it against drug target information. So we can take any of our disease genes and say, which drugs affect that disease gene? And then we can say, do those drugs share other types of biology in common, such as drug target information? So drug target information was in no way used to train our model. It was all in vitro assays that was used. But drug target information, we can ask if drugs that share a target also share projected effects on the same disease gene. And we compare this against a null model, where we just shuffle the drug target relationship network there. And we're able to show, as compared to the null model on the x-axis, these are log 10 p-value, negative log 10 p-values, and the actual significance per target on the y-axis, that there's a significant elevation as compared to this null model. And that's able to show that we're able to learn something that reflects known biology of drugs. And so in addition to looking up a particular drug and seeing what disease genes it affects, you could look up a particular target and also see what disease genes that particular target is projected to affect. So that's cool too. But a third application, now that we've shown that both we're able to predict phenotype effects and we're able to learn biology of drugs, is to see if we can go beyond drugs that are known to share biology to learn new commonalities between drugs. So we look for, do drugs actually share genes together that we wouldn't have expected? So drugs that share a target, we might expect to share the same genes, but we're able to find other relationships, other clusters of drugs that share the same gene. So this is 3 drugs that are all projected to affect CETP, the cholesterol ester transferase protein. And cholesterol ester transferase protein in the gene-based scores from GWAS is linked to high cholesterol, unsurprisingly. And of these 3 drugs, one is fenofibrate, which actually is a cholesterol drug, so that makes a lot of sense. The other 2 are not cholesterol drugs, but one is triclosan, which is an antimicrobial, but it actually acts through its effects on fatty acid synthesis, so it is plausible that it could have effect on this actual gene. And a third is flutamide, which is an androgen agonist, but flutamide is also known to target dyslipidemia. So this cluster of drugs, it's actually plausible that these all could have effects on lipid synthesis. And so our results in the paper present this whole drug network. So we're able to find these clusters of drugs that share biology, and some of them are expected and some of them are unexpected. So again, we're clustering drugs together by, do they share effects on the same disease genes? And we also have a network connecting drugs to the projected disease genes that's presented in the paper. So that's the conclusions from the publication. And here's the QR code again. So just to summarize the publication, Draphnet connects drugs to disease genes to propose the biology of drug effects on disease. And also we can use this to predict new drug indications. But because this is published, I thought I'd spend the remaining time just going over some future directions we're currently working on. So some new stuff. So the results from that paper brought up two questions. One is, how does the biology, I was talking mostly about drug indications, but how does the biology of drug side effects differ from the biology of drug indications? And the second is, can we improve this? So we took linear models pretty far, but can we do better with nonlinear models and, joining the 21st century? So for the first question, comparing side effects and indications, we were really inspired by one of the clusters that we found of drugs that were projected to influence the same disease genes. So one of these clusters includes Bepredil and Verapamil, which are two calcium channel blockers, but also includes Pimozide, that's a neuroleptic. So that's a drug given for psychosis. So of these drugs in this cluster, it really seems like two of them are antihypertensives. Makes sense that these would go together, but what's Pimozide doing there? But we actually found that Pimozide and Bepredil actually share a known side effect of arrhythmia. So Bepredil was actually no longer prescribed in the United States because of this strong side effect, and Pimozide actually has a strong warning because it can cause arrhythmia. So that was very interesting because this model was trained to predict drug indications. So it's a question, do we generally find that drugs that share side effects actually share the same effects on disease genes, even when you have a model that's trained to predict drug indications? So we can systematically address this question. If we take our model and we have our projected effects on all of the disease genes, which are from training the model to predict drug indications, we can retrain the model to predict drug side effects. So here, this drug has an indication of psychosis, but a side effect of arrhythmia. And so if we retrain the model to predict disease genes, on the side effects, do we actually get the same disease genes, or do we get new disease genes, different disease genes? So here, this is what it might look like if it's the same disease genes that are an overlapping set, or it could be distinct disease genes that were not involved in the drug's main indications. So that's something that we can ask. So if it's the first one where drug side effects are projected to occur through the same genes as drug indications, that could tell us maybe there's some hidden genetic similarity between something like arrhythmia and psychosis that we didn't know about, and that would be worth investigating, that there's some sort of way of predicting similarities between indications and side effects. If it's different, then that could be also exciting because it could suggest a new way to distinguish drug indications and side effects. The second direction that we're taking this is enriching the drug phenotype network. So hopefully this illustration is no longer too confusing to you. But what I wanted to point out here is we have a drug and we represent that drug with its profile of effects on 1,000 different endpoints. And we learn the drug's effects by connecting those endpoints to the 10,000 different disease genes through this matrix W. And this W is actually then has to have 10,000 rows by 1,000 rows, 1,000 endpoints by 10,000 columns. And if you do the math there, that's 10 million parameters, which is not impressive anymore with LLMs. But for our application, where we actually have a very limited amount of data, that's like too many parameters. So in reality, we can't learn that W. We learn a version of that W by doing a whole bunch of dimensionality reduction, which you can read about in the paper if you want. But what we want to see is, can we do something better than that by reformulating this matrix learning problem into a graph learning problem? And that's pretty straightforward in terms of like representing all of our problem in a graph. Because we already have a graph that connects disease genes to the phenotypes, and we have a graph connecting endpoints to drugs. And we could further enrich both of these graphs by connecting, for example, disease genes to each other using gene interaction networks, and connecting endpoints to their targets and targets to their target families, et cetera. But what we want to do is really the same question, but via a different method. So we can learn the edges connecting drugs to disease genes that can be fitted to predict, again, drugs' effects on phenotypes. So that's our goal using graph attention networks that we're working on right now. So that would be no longer linear models, which is, not so easy and interpretable, but I think it has the potential to not have quite so many parameters and be a bit more stable. So I just want to thank you for your attention. There's a QR code for the published paper one last time. I want to thank my students who've worked on this project and are working on the different follow-ups, Mamoon and Panos and Katie. And we're hiring if you're interested in this type of thing and these diverse projects related to drug repurposing. Lowell's a pretty good location for all types of interests. So I'll take any questions. Thank you so much.
Thank you very much. There's time for questions. If you have a question, you can walk up to the microphone. I'm maybe going to kick it off. I was wondering if you looked at all into drug combinations, if that's a possibility to model.
Yeah, I think you could look into drug combinations, but for this one, you would have to have data on drug combination in vitro effects, which we don't have. But drug combinations is definitely a topic that we're interested in the lab. So if you go to actually my talk I'm giving tomorrow, that's actually drug combination effects on cancer. And we're doing other kind of graph network models to predict drug combinations.